Hello everyone. So today we're going to be talking about eukaryotic cells. So firstly, we'll start with the introduction, where we get to learn that eukaryotic cells have a complex type of cells, and they have a true nucleus in which their genetic material is actually enclosed by a membrane. So for instance, if this is the genetic material, it's enclosed by a cell membrane, unlike eukaryotic cells where the genetic material is found freely floating in the cytoplasm. So eukaryotic cells have, the first thing that we need to learn is one, it has a true nucleus, and its genetic material is enclosed by a cell membrane. Okay. The next thing we're going to learn about is that it has what we call organelles. Okay, these organelles could be ribosomes, could be mitochondria, and so on and so forth. Now, what we notice about these organelles are they are membrane bound. Okay, so this is to show us that every chemical reaction that is occurring in one organelle does not affect another organelle because it's membrane bound. For instance, if this is the mitochondria, okay, whatever is going to be happening here, for instance, if it's tissue respiration, it's going to occur solely within the organelle of the mitochondria. Okay, so the next thing we're going to learn is that under eukaryotic cells there are two types. So we have a plant cell, and the one that I'm going to go with is the palisade cell. Okay, and the other one, this is an animal cell. Okay, so let me just use my pointer right here. Okay, so like you can see, you have this palisade cell right here and the animal cell right there. Okay, so when you look at the plant cell or the palisade cell, what you're going to notice is that it has these special organelles that we call the chloroplasts. Okay, now these chloroplasts, they contain a green pigment that refer to as chlorophyll. Okay, so when you look at a plant cell, you notice that when you look at leaves, they look green. It's because they have chloroplasts within them, and those chloroplasts have what we call the photosynthetic pigment that we call the chlorophyll. Okay, so this chlorophyll is what gives the leaves their green color. Okay, and what you're going to notice is that these chloroplasts are usually found right at the surface of the plant and beneath the surface of the leaves or the plant cells. Okay, so you notice that it's usually green at the top and even at the bottom. So this enables photosynthesis to take place, thereby enabling them efficient enough to convert light energy into what we call chemical energy, right? Into chemical energy of carbohydrates. However, we go to the animal cell, which is rich in mitochondria, like you can see right here. Okay, so this is our mitochondria, and this mitochondria, I'm going to give an example of a certain organ in the body, okay, known as the liver. So I'll draw it right here. So this is the liver in the body, and what you notice that the liver has many mitochondrial cells. Okay, and because these mitochondrial cells undergo a process of tissue respiration, so they give this so happens because there's so many processes that occur within the liver, and because there's quite a number of mitochondria found within the liver, it gives it the brownish color that we're able to see when you look at the liver. Okay, so next up we're going to talk about the cell wall. Now what you notice about the cell wall is one, it's a protective layer. So the cell wall is a protective layer. However, sorry for that glitch. However, this protective layer is not found in animal cells. You find it in plant cells. So it's a protective layer found in plant cells, and it contains what we call the cellulose and complex polysaccharides. Okay, so the major function of this cell wall include one, it gives mechanical strength to the cell, to the plant. It gives mechanical strength, okay, because it's an outer lining. So let's just go back to our diagram. So this is the cell wall, right? The cell wall that surrounds the plant cell. So it gives it strength, mechanical strength, to withstand resistance of expansion. For instance, when there is water that's coming into the cell and is trying to make the plant cell burst, the cell wall offers mechanical strength, thereby offering a certain amount of resistance. Okay, so the other reason as to why the plant has a cell wall, it controls the cell growth. So there is growth and the shape. I'm sure you've noticed that most plants have a different rate at which they grow. For instance, if the cell wall of a plant is this size, definitely this plant cell will not grow beyond this. Okay, but there are certain leaves that are really big. For instance, if that let's take for instance the banana leaves, right? So the cell wall offers growth and the shape of this, because you know that this is rigid in such a manner. Okay, then it also provides intercellular connections using its pores. So it provides intercellular connections. Okay, because the cell wall has different a variety of pores within itself. Pores are simply tiny holes through which certain substances are able to go in and out of this cell. Also, the cell wall reduces water loss and also risk of infection. You know, it's not only human beings that have infection, but even plants are able to get infection. Then it enables the long distance translocation. Okay, translocation of materials. Okay, so what you notice, if this is your xylem, your phloem vessels, right? So there is transportation, could be these are water molecules. So there's a cell wall that lines this xylem and phloem. It gives it the structure. That way, all these materials are able to pass through and not really leak out like that. It gives it strength in order to transport these materials. An example I can give is of a hose pipe. When you turn on the water, water is able to easily flow. Unlike where there is a loophole, there is a leak somewhere, you find that water, it will reach quite alright, but there are some parts that won't efficiently reach because of those pores that are there. So this cell helps to transport those molecules from the roots through the xylem and the phloem all the way to the branches in order for the plant to be nourished. The next thing we'll talk about is the plasma membrane. So the plasma membrane consists of 40% proteins and 60% phospholipids. Okay, so an example of a phospholipid, for instance, this is your phospholipid, right? It has a phosphate group attached to a lipid molecule. So what happens is that this is what will be happening, right? So within itself, it functions as selective permeability, because there is one part, the phosphate group is water loving, which is hydrophilic. So this is water loving, okay, which is hydrophilic, okay. The other end is hydrophobic, okay, meaning it's water hating. So because of this, certain substances are unable to pass because this cell membrane is highly selectively permeable, thereby controls information flow between cells and the environment. It has through its specific receptors of energy. It also helps in the conversion of ATP during photosynthesis. Now, ATP is like the energy currency of the cell, so it's also being used for production, okay, production of ATP during photosynthesis, and also oxidative phosphorylation, okay. So this is what the cell membrane is being used for. The next thing we're going to talk about is the nucleus. So the nucleus, one, is the largest organelle. Let's go back to our image. So this is the nucleus right here at the center that you can see. So this is the nucleus, and it's the largest cell organelle that's found in almost all eukaryotic cells. So this nucleus contains what we call genetic information, okay. It contains DNA, which is used to control all cellular metabolic activities, okay. Now these metabolic activities, I'm just going to mention a few. So you have for instance, DNA replication, okay. So there's so many metabolic activities that occur. So the nucleus is surrounded by a membrane that we call nuclear membrane, okay. Remember we said that eukaryotic cells are membrane bound, okay. So it's got a nuclear membrane around it, and this nuclear membrane contains what we call a nucleolus, okay. This nucleolus is this one right here, okay. This is the nucleolus, which is part of the nuclear membrane. So when you look at the nucleus, the function of the nucleolus is that it manufactures ribosomal RNA. So this ribosomal RNA is produced by the nucleolus. So abbreviation is RNA. Okay. So the nuclear membrane has what we call nuclear pores. Okay. Like you saw, there are big spaces that were there. Let's just go back to the image. So these big spaces, these are the pores of the nuclear membrane. Okay. Now these pores are there for a reason. And why are they there? It's because they allow exchange of substances between the nucleus and the cytoplasm. So when you look at this nucleus is within this cytoplasm, right? This is the cytoplasm that you're able to see. So because of these pores, once DNA is formed, it's able to come out of the nucleus using those pores in order to undergo further modification, okay. All right. Most of these things are seen in processes that we call transcription, where the DNA is encoded in order to make RNA, tRNA, and so on and so forth. Okay. So then within the nucleus, it has what we call the nucleoplasm, and it's a fluid containing enzymes, genetic material, and so on and so forth. So the nucleoplasm is right inside the nucleus. So that fluid has many enzymes through which those enzymes help in those metabolic reactions that we talked about. The next thing that we talk about is the vacuole. Now the vacuole is an organelle that is present in all plant cells. Let's go back to the plant cell. So this is our vacuole right here. Okay, so the vacuole is simply a space that is found, okay, and it's bound by a membrane that we call the tonoplast. So this vacuole is filled with water. So let's just write down what's found in the vacuole. One, it's filled with water. It's filled with water containing inorganic and organic molecules, including enzymes. So these enzymes are also found in that space, and it's involved. So what's the function of the vacuole? It controls what we call turgor pressure. Okay, it controls turgor pressure. It maintains the pH, and it's for removal of harmful substances. Okay, so these are the things that occur. Certain waste products are removed from the plant cell using the vacuole. Okay, so the vacuole size or the shape varies depending on the cell type. Okay, the type of cell and the function. Okay, so in there, if you see that this space of the vacuole is like this in this plant cell, it won't be the same for another plant cell. Maybe it could be circular, could be oval, depending on the function of that cell. Okay, okay, so this is the vacuole, and remember that we said it controls the pH. So because it has fluids, and within those fluids we have enzymes and whatnot. So the pH could be acidic, basic, and what controls that is the vacuole. And for the turgor pressure, this turgor pressure is also helping hand in hand to prevent this cell from bursting. For instance, if water is going into this cell at high pressure, the vacuole is able to resist that pressure. So the next thing we're going to talk about is the rough and the smooth endoplasmic reticulum. So I'll take you back to the image. So this is the endoplasmic reticulum. So this one is called the rough endoplasmic reticulum because it has ribosomes attached to it. So the rough ER, which is endoplasmic reticulum, has ribosomes. Okay, whilst the smooth ER, this one has no ribosomes. Okay, so this is why they're called rough and smooth endoplasmic reticulum. So the rough endoplasmic reticulum, which is the rough ER, this one is used to transport proteins. Okay, you know that protein synthesis occurs within the ribosomes, which we're going to talk about later on. But after they are produced, what transports them is the rough ER. Okay, it transports the proteins, while the smooth ER, on the other hand, this one transports lipids. Okay, it's involved in the synthesis of lipids. Okay, so in the liver cell, both the smooth ER and the rough ER are involved in the removal of toxic substances. So this is the liver. So they're both involved in removal of toxic substances. Okay, so this is the function of the rough and the smooth endoplasmic reticulum. Next thing we're going to talk about is the Golgi apparatus or Golgi apparatus. So this organelle is found in all eukaryotic cells, and it contains a pile of sacs. So for instance, this is a pile of sacs that are stacked together right here in this image that you're able to see. So it contains a pile of sacs that are stacked together, and they're called cisternae, and they also have smaller Golgi vesicles. So let's just write that down. So it has what we call, the sacs are called cisternae. Okay, and it has small Golgi vesicles. Okay, So the function of the Golgi apparatus is that it modifies and transports materials. Okay, so there is modification, right? If you're modifying something, it's like you're cooking rice. Okay, if we say I'm modifying it, it means it's already made, but you're just making a few changes to it. So it modifies and transports materials. Okay, so it modifies and transports materials. Okay, and materials particularly in the secretory cells, such as the cells of the pancreas. When the pancreas produces certain hormones, it will modify them and transport them. This is what it does. And also it's involved in lipid transportation from the smooth ER to the lymphatic system. Remember, the smooth ER was involved in lipid synthesis, right? So what happens is that when these lipids are synthesized, the Golgi apparatus is then going to transport them right into the lymphatic system. Okay, next thing we are going to talk about is the chloroplast. What's the function of the chloroplast? So we know that the chloroplast is found solely in plant cells. Like you can see here, these are the chloroplasts that we talked about, and we said that mostly found at the surface of the plant and beneath the epidermis. So the chloroplasts are organelles that are used for the process of photosynthesis. Okay, and so why are they being used for photosynthesis? It's because they contain a pigment that we call chlorophyll and the carotenoids. So we have this pigment that we call chlorophyll and carotenoids. Okay, so this is what is formed. So these are mainly found in the palisade cell or the green leaves. Okay, and they are surrounded by two membranes. Okay, they are surrounded by two membranes. Okay, it's like a double membrane right there. Okay, which contains the chlorophyll. So wow, which we drew here. So these are the chloroplasts that are surrounded by two membranes. Like we can see, we have two membranes that surround them. Okay, so these chloroplasts, we said they contain chlorophyll and photosynthetic enzymes. These photosynthetic enzymes are being used in the process of photosynthesis. So the membrane consists of many sacs that we call the thylakoids. We have what we call the thylakoids, which form the sacs that we call the grana. So these are the membranes. They have what we call the thylakoids. For instance, if these are thylakoids, and within these thylakoids is what we call grana. Okay, these grana are interconnected by lamella. Okay, so if this is grana, okay, they're interconnected by what we call lamella. Okay, so these are the chloroplasts. The next thing we'll talk about is the mitochondria. Now, we know that the mitochondria is the powerhouse of the cell. So the mitochondria has, it's an organelle that is present in all eukaryotic cells and is the major organ that we use for aerobic or oxidative respiration. So aerobic or oxidative respiration. Okay, so this is the powerhouse because this is where energy is being produced. The famous ATP is being used, and we said many of these mitochondria are found in liver cells as well as muscle cells. Okay, muscle cells. Okay, so each mitochondria is bound by two membranes. Just like the chloroplast, it's got two special membranes. There's one in the inner and also one in the outer. Okay, the inner membrane is semi-permeable. Okay, so this one is semi-permeable. It doesn't allow all substances to go in. Okay, whilst the inner membrane is semi-permeable and it touches the cytoplasm fluid, like you can see here. Okay, so it touches the cytoplasmic fluid where it's folded. So these that you saw that are folded, these are what we call cristae. Okay, these are folded. So the cristae increases the surface area. For instance, if you're asked which one has a larger surface area, okay, you have one straight one and one super coily, okay, fitting in the same space. The answer is going to be this super coily, because when you stretch the super coily one, cristae is going to be this long. Okay, so meaning there is more surface areas compared to this straight one that's right here. Okay, so that's why it has a cristae. So the outer membrane is permeable to many membranes. The inner one was semi-permeable. The outer one is permeable to many molecules, including even the large ones. So although these so the mitochondria has its own DNA. Okay, it's a special kind of cell. It has its own DNA. However, it can't reproduce independently on its own. It needs the nuclear DNA for its production. So in as much as it has DNA, it cannot replicate itself or produce itself. The next thing we'll talk about are the ribosomes. So the ribosomes are very small organelles, like you saw in the beginning. They're quite very small. So these are the ribosomes. They are very small organelles that are found in large numbers throughout the cytoplasm, like you saw. So they're found in both plants and animal cells. So what's the function of these ribosomes? So these are sites of protein synthesis. Okay, so these ribosomes are like the factory where the proteins are made. So in eukaryotic cells, we have the ribosomes they are defined by a number that we call the ATS. So the ribosomes are formed by what we call subunits. For instance, it has a large subunit and a small subunit. However, these subunits are they're not additive. Okay, so this S sound denotes Svedberg units. So these are not additive. So ATS, it means the large subunit is 60S, the smaller subunit is 40S, and these do not add each other to give you ATS. Okay, so the same. So these are the ones that are found in eukaryotic cells, while in prokaryotic cells it's 70S. So you have the large subunit being 50S, whilst the small subunit having 30S. So these subunits, they come together in a process called protein synthesis. Okay, so the large subunit comes with mRNA, with tRNA, sorry, that is bound, and the smaller subunit with the messenger RNA. And when this occurs, all the genetic information that's encoded on the messenger RNA is translated into what we call proteins. Okay, so this is what occurs. So these ribosomes can be found freely floating in the cytoplasm, like you saw here. These are freely floating. Other ones can be bound to the ER. Okay, those are the ones that we're calling the rough endoplasmic reticulum. Next up, we'll talk about the microtubules. So these microtubules are actually cylindrical. Okay, they are really cylindrical. They look something like this. Okay, so these organelles are cylindrical organelles. They're made up of a protein that is called tubulin. Okay, tubulin are microtubules. So these are involved in a number of cellular processes such as formation of centrioles and intracellular transport. So let's write it down. So you have intracellular transport. Okay, you also have making up of the cytoskeleton, as well as the spindle fibers. Okay, spindle fibers, these can also be found in muscle cells. If you've seen the muscle cells, they have what we call the spindle fibers. Okay, so within them they have what we call the microtubules. So next up, we'll talk about the centrioles, which we said are produced by microtubules. So these centrioles are small hollow cylinders that occur in pairs called a centrisome. Okay, they occur in pairs like this. They are small hollow cylinders and they occur in pairs, and this is what we call a centrisome. Okay, so in most animal and lower plant cells, this is where you find the centrioles. So at the beginning of nuclear division, the centrioles replicate themselves. So there is what we call nuclear division. This is where the centrioles replicate themselves into two pairs and separate to opposite poles. Okay, these are the centrioles. They separate to opposite poles. One goes the other way and the other one goes to the other way of the spindle fibers, in which the chromosomes are well aligned. So this occurs during nuclear division, meiosis. So sorry, just to take you back, let me make a correction. So these spindle fibers are different from muscle fibers. Okay, let's just make that clear. The spindle fibers, you see, these are, if this is a genetic this is your DNA, we have what we call the spindle fibers. These spindle fibers are the ones that pull away the DNA towards each pole. Okay, this is during nuclear division. These are what we call the spindle fibers. Okay, those are the ones that were being formed by the microtubules. So this is what happens. Those are the centrioles that separate during nuclear division. Thank you so much. I hope you enjoyed this lesson.